Good morning, biology class. Welcome back. We're going to go ahead and make a graph today. This should be your lesson for Wednesday, March 18th. We want to keep this as organized as we can. One Edmodo post per day. That's your whole lesson. Now, today our goal is to make a graph. It should look something like this. It's not going to look exactly like this, so we're going to go ahead and get rid of this. But what we're going to do is create an assignment for this whole lab report thing on Edmodo, so you can turn it in on Edmodo, and that's really important for you guys to know. If you need to email this to me because Edmodo is being bogged down by the millions of kids using it, that's totally fine. If you are handwriting this and you want to take a picture of it and send it to me with your cell phone, you can do that too. Just take a couple pictures so I can actually zoom in and read it. Not going to be that convenient, but is totally doable. So that's an important thing to note. So we said our due date was Friday on this. I know that you guys have been jumping around to many different emails and different Edmodo posts for all of your teachers. So I'm comfortable extending this into the weekend. If you want to turn this in Saturday, Sunday night, I'm fine with that. But let's really not push it any further past next Monday because we do want to keep progressing with this. So anyways, I told you last time that we're going to make a video on how to make a graph, okay? The best way to make a graph. This is our hypothetical paper that we put together last time, and we're on the results section, and we wrote out a couple sentences describing how many colonies we grew in what section. Now, it's important to note that if your computer does not have Excel, you can pursue online graphing websites that can do the same thing. But if your computer can't screenshot easily, then doing them on an online graphing portfolio or website might be really difficult because you might not be able to get it into your Word document. So worst case scenario, give yourself a big space, and here you have your sentences describing your colonies, your results, okay? And underneath of that, you're going to put a graph. And so on and on and on these sentences go, and then we're going to put a graph. And so our graph is going to be figure 1, right? That's our graph. Now, if you want to put a space down here like this and hand draw this graph with colored pencils or something, be my guest. You will not be docked for hand drawing a graph if that's what you need to do. As long as it has a title and labeled axes, you're going to be in great shape. So if you have to do that and your computer doesn't have Excel, or there's multiple kids in your household sharing the same computer because the schools are closed, by all means, color yourself a graph. Draw it with a pencil. That's totally acceptable and will not affect your grade. Okay, remember the goal is learning, not hoop jumping. But for those of you who have Excel, go ahead and open up Excel and make something along the lines of this. Now we have location. And this one I'm doing this plate right here, which is different parts of the body, right? Foot, finger, arm, mouth. Foot, finger, arm, mouth. Straight across the top. Now, I did number of colonies on this because the graph is a little more exciting. And that I don't know if that really addresses my hypothesis. I'd have to go look back at my hypothesis. If my hypothesis was the most diverse bacteria, which I think it was, we should change this. Mouth, no bacteria. Foot, lots of bacteria, but they're all the same yellow speck. So the odds of that being the same type of bacteria is really quite good. So in diversity's sake, we should honestly put a 1 for foot. Right now I have 35 for number of colonies, but if we're going to put the number of types of different colonies, it should really be 1. So let's go ahead and change that, because I think the hypothesis I wrote with you guys yesterday was about diversity, not the total number of colonies. So let's go back here and let's go number of different colonies, and that's actually addressing our what would you call it? Our hypothesis, right, of diversity. So let's go into that. Foot, I'm going to say 1. There's a lot of yellow spots, but they're all the same yellow spot. So let's actually change that 35 to 1. Now, if your hypothesis was what's going to grow the most bacteria, then you want to go with the 35 and count your colonies. Does that kind of make sense? Our results need to match our experimental question at the beginning. okay? My experimental question wasn't, does more bacteria grow on a foot or a finger? My experimental question was, which place is the dirtiest, has the most diverse bacteria? And there's only one type here. So the finger, being all like whitish gray, right, is also probably one type of bacteria. So let's go back in here to the finger part, and let's make that just one as well. Now the arm, this is where we actually get something kind of cool. We have yellow, we have orange, we have red, and we have a white mold. So 1, 2, 3, 4 different types of bacteria easily. So let's go back to arm, and instead of 14, we're going to make that 4, okay? Now, your experimental question does not have to be, what's the most diverse sample? Yours can be the number of bacteria, and you can go by counting the colonies. That's fine. Just make sure it addresses the question you want to address. So we're on Excel. We have this created. How do we turn this into a graph? Well, on Excel, it's very, very easy to do. You highlight the area you want to work with, and you go over to Insert, and it gives you all of these options, okay? Now, we're going to go with a pretty simple bar graph. That's probably going to be our best bet for this. So come over to this button that says Column. By the way, if you're on office.com, your school Outlook Excel, and you don't have the ability to screenshot, you need to abandon what you're doing. I'm so sorry, but unless that's changed from last year, you cannot take a graph from the free online Excel that you all have access to through your rcchristian.org account and paste that graph into the free Word Online that you also have access to. Why that is that way, I will never know. It's ridiculous. So if you're on an Excel program that's downloaded on your family's computer or downloaded on your laptop, then proceed along with me. If you're not, abandon ship now. Get your colored pencils out. Make your graph with colored pencils and get a great grade, okay? Still make the graph in the same format. So by all means, watch to see what the good graph looks like. But don't follow along step for step on Excel, only to not be able to move it over to Word. That bothers me, and I'm sorry. I don't work for Microsoft. There's nothing I can do for you. So anyways, we have a highlighted area of what we want to do. So all we're going to do now is come down to this first graph with bars that are in 2 dimensions and click it. And you're almost done. It's that easy. Isn't this cool? I might make this bigger just so you guys can see what we're going to do with this here. I like that better. Okay, so what we've got is a very boring graph with a y-axis, an x-axis, and the title, which the title actually worked out to be great because it's the number of different colonies. That's really not that descriptive, so let's change that. Let's change that to what? This is foot and arm. Number of different bacterial colonies. I like that better. On body surfaces? Why not? I like that. That's fine. Okay, that's a very descriptive title, right? People read the graph and they know exactly what's going on. There's no question there. I think our gradient of 1.52, 2.53 is probably not necessary as far as how this is broken down, but that's fine. I'm going to show you the important elements. So what I want you to do now is to have a good graph. For those of you kids who are coloring with the colored pencils right now, I'm talking to you too. To have a good graph, you need a title and a labeled axes on both the X and Y. So on Excel, all we have to do is you're going to come over here to add chart element, okay? And you're going to hit axes titles, sorry. And let's do the horizontal one first, why not? So what is this? This is location. Location of, should we call it sampling or samples? Let's go location of sample. I like that. So when somebody reads this, foot, arm, finger, mouth, what is this? Oh, this is the location where they got their sample from. Probably already knew that, but we want to have labeled axes anyway. So now come down, axis title again, and we're going to do the vertical one. Now this one kind of messes with me because you have to type sideways, but it totally works. So what are we measuring here? We're not measuring the number of colonies, we're measuring the, what would it be, the number of different bacterial colonies? So yeah, number of different colonies. So here we're going to write number of different colonies observed, grown, I don't know. Let's go observed. Counted? Ooh, there's so many options. I don't want to overthink it. All right, now we're done. We leave the graph alone. Number of different bacterial colonies on body surfaces. We have a descriptive title. We have a labeled y-axis. We have a labeled x-axis. These represent well the colony numbers that we have here. So you're done. Like, I don't know if we took away all the jibber jabber that I was going on about how much Excel stinks working with Word Online. Like, you can do this in like 6 minutes, maybe less. I don't know. Okay, so now all we have to do, this is a really cool part, right? Results, on and on with your sentences. The arm grew 4 different bacterial colonies. All were circular, orange, yellow, white, and a filiform mold, right? Period. And on, that's where all these sentences go, is right up here. Now, figure 1, you literally click over here, grab your graph, pull it off of this, put it in your Word document, and let it go. Now we jump to our Word document, and you did it, right? You're done. If you want to change the size, you can change the size. If you want to be a little more accurate with APA and underline figure 1 or put it in bold or, you know, whatever, you can do that if you want to. It doesn't bother me, right? And we're done. Like, we did it, okay? So yesterday, or even before that. I know some kids left school with their lab report halfway done, so it's kind of funny to be doing this, but introduction should already be done. Materials and methods should already be done. Results, what you could do in handwriting or typing, should be done. So now results is done, and discussion and conclusion should be done, right? The arm is the most diverse because it's, I don't know, exposed to more air than the other body parts. I don't know. Pick some sort of explanation for your particular results, because I know your results make a lot more sense. My results are garbage. I assure you my mouth is not sterile. That's ridiculous. So whatever. You got to work with it though, right? That's the results. Okay, so now all we have to do, if you've been following along, is write an abstract, and you're done. And so the abstract, if you've lost your syllabus or notes, should be what you did, how you did it, your results without all the data, and what they mean. Okay, that's really it. A really short paragraph that at a glance tells somebody exactly what was done, what the results were, and basically, is this experiment worth their time reading or not? And so let's jump right into this. I'll just type this along. Again, do not copy my abstract. Make your own abstract. But for the sake of formatting, we'll knock one out real quick, shall we? Past tense narrative, avoid I, me, we, us. Let's see what we can do. An experiment was conducted to examine which part of the human body had the most diverse bacteria. Yeah, let's stop it there. That looks good. Okay, so why did we do what we did? And okay, so now we got to do, what did we do? Okay, what did we do? That's our reasoning. Should we put a hypothesis in there? Abstract? Nah, we'll leave the hypothesis in the introduction. Put your hypothesis in your introduction. That should not be bold. I apologize for that. I'm tempted to put a hypothesis in there because it just flows so well, but we shouldn't. Let's stick with what we're supposed to do. Cotton swabs. We'll get away from that copyright infringement with the Q-tip name, shall we? Cotton swabs were used to transfer bacteria from, let's see, a foot, arm, finger, and mouth to an agar plate. The plate grew more colonies from the arm than from any other surface. This shows, now this is the hard part, right? What does it mean? Golly, which is ridiculous because it means my arms are dirty, dirtier than my mouth, which doesn't make any sense. So I don't even know how to explain this data. That's tough. I don't know the answer to that. Let's think. An experiment was conducted to examine which part of the human body had the most diverse bacteria. Cotton swabs used to transfer bacteria from a foot, arm, finger, and mouth to an agar plate. The plate grew more colonies from the arm than from any other surface. This shows that. I don't know. This potentially shows that the surface area and frequent contact to desks, laptops, etc., create an ideal environment for diverse bacterial growth. Okay, and I'm tempted to say, against the hypothesis, refuting the hypothesis that the mouth would be more diverse. I'm tempted to say something like that, and I really don't feel like I should. It's an abstract, just the facts, ma'am. What'd you do? How'd you do it? What are the results? What do they mean? And because of my goofy results, saying what they mean is actually a little bit challenging. But really, your abstract does not need to be any longer than that. A succinct writer, a few good sentences for a lab of this complexity, and you're done. And so you guys should pretty much be done. So now save that document, go into Edmodo, and there will be an assignment. And in your biology class, there will be an assignment with a due date. You go in there, you upload it, and I will give you comments and feedback, and you guys will be in great shape. So tomorrow we're going to start viruses. Let me know in the comments of this video if you're interested in trying a Zoom. It's weird, I think, to just talk to a screen. It's weird to not see you guys and for you guys not to see me, and for you not to have the ability to raise your hand and ask questions. That's driving me crazy. So if you're interested in it, drop that in the comments. Zoom is like a Skype or a FaceTime for a big group, and you can see my notes, you can see my face, and I'll sit in my kitchen with a cup of coffee, and you can listen to a bio lecture. And then what we'll do is we'll record the Zoom meeting, and we'll post it to Edmodo just like I'm posting these videos now, so that if you can, let's say we set the Zoom meeting up. We'll see what time works best for you guys. Let's say we set a Zoom meeting up for 9:30 in the morning, right? If it works with your schedule, you jump on at 9:30 in the morning, and you can stop me, ask questions, be part of the discussion, and not have to do this, throughout the day at random times. But if you can't do 9:30 in the morning, right? You have some sort of conflict. Well, as soon as the lecture is done, I'll save it and I'll post it. And then you could come back and watch it just like you're watching these videos right now. So if you're interested in that, respond to this video with a comment that says yes for Zoom noon, or yes for Zoom 9 a.m. or no for Zoom, I like the way we're doing it now. Let me know, and I think it would be cool for you guys to be able to ask questions, especially as we get into additional lectures, which starts tomorrow morning. So let me know, and if you want to give it a shot, I'm definitely up for giving it a shot. So anyways, that's really it for this lesson. Complete your lab report. This thing is due at the end of the weekend, okay? Sunday night. I'm giving you extra time, more time if the family computer's crowded right now, all right? Sunday night. If you have questions, I'm trying to check my email right now a dozen times a day. If you have questions or something's there's some sort of emergency or something, and we're going to be doing this online school thing potentially for a while, you can text me or give me a call, okay? I don't like giving my cell phone number out to students, but I tell you what, since we're in this weird situation, here's my cell. 605-786-3685. Drop me a text if some emergency happens or whatever, or a question that you have. Just do it before Sunday night at 11:50 when you're uploading your lab report and you're like, the assignment's closing in 9 minutes. Well, don't wait till Sunday night. Come on, But I'm trying to give you a few extra days. I'm trying to be flexible with you. So if you need something, email me, Edmodo, message me. Drop me a text, give me a call, whatever. I'm here to help you guys. I apologize for the format of the digital schooling thing, but bear in mind the goal is learning, not hoop jumping. And if we focus on that, we're going to be fine. We can learn a lot even if we're doing it digitally, and you guys are going to do great, okay? So let me know if you have any questions, and I will see you guys tomorrow. Thanks.